Good morning, everybody. My name is Jennifer SanMiguel, and I'm a study director here at the Jackson Laboratory in the Rare Disease Translational Center. And I'm excited to share with you our SLC6A1 lock-stop-locks mouse model today. So this project nomination came to us through Stephen Gray and Amber Freed, and they asked us if we could make a mouse where we went from no expression of SLC6A1 to an inducible revertant of the wild type gene. So what we did is we inserted a lock-stop-locks cassette, which carries a strong splice acceptor and viral termination sequence, into intron 6 of the endogenous mouse locus. So pre-Cre, this animal carrying this stop cassette is essentially null. And then in the presence of Cre, either genetically or mediated through AAV, we can get recombination of this cassette. So this termination sequence goes away. And now we've reactivated the wild type transcription of the gene. So this is basically a rescue allele. And the benefits of this allele are exactly what you would think. Now we have the ability to assess when to reactivate wild type SLC6A1 and where to reactivate it. Do we need to reactivate it in all the cell types where this gene is normally expressed, in some of the cell types? And these are two of our long-term study goals. But today I'd like to talk to you about the molecular and phenotypic verification that we've been doing on this allele to give you a little bit more of an idea of what this mouse is looking like. So we started by doing het-by-het breedings to see what we got, and we've validated that we have equal ratios of male and female coming from this cross, which is great. And we're also seeing nice Mendelian ratios coming out of the het-by-het cross. However, when we tried het by homozygous crosses or home by home crosses, most of these pairs were unproductive. And so moving forward, we'll be sticking with the het by het cross. We've also taken some of these animals down to prepare tissues for molecular validation. And so we weighed these animals ahead of time before the takedown, and we were also able to look at the body weight. So this is some 9-week body weight data with females on the left-hand side and males on the right. And in order, it goes from wild type, heterozygous to homozygous. And you can see in the females, the homozygous females are trending towards reduced body weight. And this is what we would expect for an SLC6A1 null mouse. And the males are showing significantly decreased weight compared to their wild type or heterozygous litter mates. And so this is a really nice early functional validation that this allele is working correctly. So in terms of molecular, we designed an assay to look at RNA expression, where the assay sits in exon 7 and exon 8, which is downstream of this lock-stop-locks cassette. And what that means is in our pre-Cre mice, which are null, there should be no transcription past this stop cassette. And so this assay will turn up either undetermined or a blank band on a gel. And then after recombination, where wild type expression has been reactivated, we should indeed see expression downstream in exon 7 and 8. And so we took a first look at this just using regular PCR to validate our primers, and we looked in both liver and brain. In liver, SLC6A1 in mice is not expressed, and you can see across different sexes, male, female, across different genotypes, either homozygous or wild type, and then in pre or post-Cre animals, there's no expression of SLC6A1, which is expected in our negative control tissue. In brain, in both female and male pre-Cre homozygous animals, there is no SLC6A1 expression, which again confirms that this allele is working properly. Pre-Cre, there should be, the stop cassette should be working, and we're not seeing any downstream expression. Our positive control here is a female wild type pre-Cre mouse, doesn't have the lock-stop-locks cassette, and indeed has wild type expression. You can see a nice band here. And our female homozygous post-Cre animals are also showing expression of the wild type band, showing that the allele is able to be reactivated upon Cre expression, which is great. We've also looked at the copy number of our lock-stop-locks cassette. We want to make sure that it only got inserted once per allele, and so we can assess that by using digital droplet PCR or ddPCR, and we can compare that to another gene that's normally in the genome. So RPP30 is our reference allele, which is 2 copies per genome and is represented in this gray bar. So you can see in our SLC6A1 wild type, there are 0 copies of the transgene, which is what we expect. In our SLC6A1 heterozygous, there's one copy, so one allele carries a transgene and one doesn't, which is correct. And then our SLC6A1 homozygous animals carry two copies of the transgene, so two copies per genome, and that's exactly what we want to see. So now that we have this nice molecular validation, we are moving on to our phenotypic validation. We've been actively breeding up these mice, and we are actually mating our cohort breeders right now to get ready for our phenotypic validation. So we'll be just validating the model to look for phenotypes that were present previously reported in SLC6A1 knockout alleles. So we'll be looking at things like survival and body weight, and I've already shown you that we're seeing reduced body weight in our pre-Cre null animals, which is great. And then we'll be assessing things like behavior and memory, so looking at the nest building assay, open field, and contextual fear conditioning, as well as neuro and motor function. So we'll look at hind limb clasping, rear leg movement, and assessing tremor in these animals, as well as looking at motor coordination using the Broderod assay. And then we're really excited about our collaboration with Dr. Paz and her research postdoc, Yulia, at the Gladstone Institute, where they will be doing the EEG characterization on these animals and also validating some of the same assays at their own institution. So we're looking forward to working with them closely. Lastly, we've made these animals publicly available over the summer, so you can buy breeder pairs at MMRRC. And this is the stock number 71548, or you could go to the website here or scan the QR code. It'll take you right there. And we also love to collaborate. So another collaboration that we have ongoing is with Henry Lee at Children's Hospital Boston. And so if you're interested in working with these animals, feel free to buy some breeder pairs or reach out to me at jennifer.sanmiguel@jax.org, and I'm happy to chat about any questions about the model you have or any collaboration ideas you may have. So with that, I'd like to thank everyone in the Rare Disease Translational Center, led by Kat Lutz, as well as our collaborators here at JAX, so Steve Murray, Kathy, Stephen, Ben, Yaned, and Amir, who were instrumental in generating these mice and characterizing them molecularly, as well as our external collaborators on this project, Stephen Gray, Henry Lee, Jean Paz, and Yulia, as well as our scientific services and our funding. And with that, thank you very much, and I look forward to hearing from you.